Our next guest speaker, I have the great pleasure of introducing Dr. David Dedi Meiri, who is a Principal Investigator at Technion Israel Institute of Technology, Laboratory of Cancer Biology and Cannabinoid Research. Now, I do have a little bit of a bio, but I've just been told by Lucy that it's a little bit outdated. Should I read it anyway? May as well, hey? So you can say tick, no, you might even be able to get up here and say, okay, that bit is just so history. You can tell us all. All right, here we go. Now, this is what I collected on you. Dr. Meiri conducted his postdoctoral fellowship at the Ontario Cancer Institute, where he focused on the role of GEF-H1 protein in tumour invasion and metastasis. During his postdoctoral studies, he expanded his knowledge in human biology and cancer pathogenesis, and succeeded within a very short time frame to receive worldwide recognition for his research. How am I going so far? Yeah, all right. Okay, awesome. Okay, I'm going to keep going. Upon completion of his, I'm feeling bolstered by your affirmation, thank you. Upon completion of his postdoctoral fellowship, Dr. Meiri took a position at the Technion Israel Institute of Technology, where he heads the Laboratory of Cancer Biology and Cannabinoid Research. Presently, his lab investigates the therapeutic potential of phytocannabinoids, the unique active compounds of the cannabis sativa plant, and the anti-tumour effects of cannabinoids. Is that hopelessly outdated, or is it sort of, am I in the last 5 years at least? Okay, all right, excellent. You're right, well, thanks. I feel heartened by that, I think. All right, well, I'm going to keep going and see if this is also part of your prescient experience. Dr. Meiri collaborates with cannabis growers, clinicians, major manufacturers and distributors of medical cannabis for the purpose of revolutionising cannabis treatment. He is operating the Cannabis Database Project, and his lab is currently involved in 8 clinical trials covering diverse aspects of cannabis treatment, such as colon disease, pain prevention, cancer treatment and epilepsy. He's highly involved in governmental regulations and is a residing member in several Israeli Ministry of Health committees which seek to advance the fundamental understanding of optimal cannabis usage and minimisation of adverse side effects. Please welcome Dedi Meiri, who will provide us with insights from his presentation, a research update from Israel. Welcome.
After such a long introduction, I don't need to speak anymore, I think. I think there is a point that you need to stop telling about your postdoc because it's ancient. Thank you very much for having me here again. It's an honor and a pleasure to be and to speak face to face after 2 years of lockdown. I'm sorry that the other speakers from abroad didn't came. I'm quite surprised. But again, I think it's extremely important, these face-to-face meetings and talks. It's not the same thing as doing a Zoom. I did, I think, 4 talks in the previous years in Zoom. I can tell you that speaking in Australia, I wake up at 3 a.m. in the morning, not wearing real shorts, and giving a talk and half an hour after that going back to sleep. So it's not the same. So cannabis, I don't think that I need to do a long introduction anymore about cannabis, but I think there is few things that I see that are not really understandable about cannabis, and I think it's extremely important to understand the plant in order to understand how we can treat real patients. I will put time that I will know how much I can spend. Never mind. Just a moment. Okay. So, cannabis. Cannabis, it's a unique plant because it's harboring unique compounds called cannabinoids. I think everybody in the audience know that. But when I'm asking people where cannabis is growing in the nature, there are many answers, you know, Nepal, India, Siberia. And the truth is more complicated, because if I will ask you about dogs, about poodle dog, where it's growing in the nature, so actually, and all of you will answer that it's not really growing in the nature, right? It's something that we, habitat, the people. So we started from these wolves that started to go around people that started to hunt, and we breed them, and we got these after a few years, right? Cannabis is a poodle. For 10,000 years, we are breeding that. There is no cannabis, sativa, indica. These are bullshit, right? Like, who have, all the breeders here, who have 100% sativa or indica, 70%, 30%, as somebody that did a PhD in plant molecular biology, what the meaning of 70%, 30% indica sativa? There is not such a thing. So cannabis is a unique plant because people breed it in a way that will harbor a huge amount of cannabinoids. If we are looking on other plants that harboring cannabinoids, there is 0.1, 0.3, 0.3. Cannabis can hold 30%. It's because we breed it. It wouldn't survive in the nature, or in few generations it will lose this amount. These are sticky molecules that probably creating to stick the pollen from the male to the female, and this is the reason it's on the flowers. But we breed it in a way that it will harbor a huge amount of these unique molecules, phytocannabinoids. And the reason people did it, because these molecules affecting our body very powerfully, because it's affecting our endocannabinoid system. And again, we need to understand a little bit more about the endocannabinoid system. Every cell in our body, harboring the endocannabinoid system and producing endocannabinoids. So it's actually a system that communicates with all other systems in our body. Our body produces molecules like the plant. And this system is a homeostatic system in most of the cases, 80% of the cases. It's balancing the other system, whether we are talking about appetite, sleep, pain, immune system, our heart beating, glucose metabolism, all of these being controlled by the endocannabinoid system. It's a mild system. We have a severe system in our body, and near that, the endocannabinoid system. How we feel pain. We have the opioid system. If I will cut my hand totally now, the body won't play with the endocannabinoid system. It will operate the opioid system in our body. But there is a big penalty. We will be totally out. Our heart beating will reduce. Very severe action that the body will do to survive. But now if I will just hit my hand like that, after 5-7 seconds, this pain will release. And it will gone because the endocannabinoid system, very locally, very specific, a specific cell will release specific endocannabinoid that reduce this pain. But this is very locally, very fast, 7 seconds, active, not active. And this is the endocannabinoid system in all our body, and I need 2 hours in order to go system by system, what is the immune system, how it's reacting in pain and sleep. But cannabis is affecting this system. It's actually a communication between cells. One cell sends a signal to another cell, reduce pain, increase appetite, reduce insulin secretion, and more. The molecules are the endocannabinoids. And in the other cell, there are receptors, there are proteins that capture this molecule and send the signal. Cannabis was just unlucky to produce similar compounds, and therefore, it's overpass what the body knows to do by itself. When we're smelling a barbecue, small molecules of the barbecue entering our nose, going to our olfactory system, signaling to a cell that produces a specific endocannabinoid, going to the liver that create insulin. There is feedback loop to increase appetite. Cannabis is just overcome that. Instead of smelling something from outside, one molecule will bind the receptors and will increase appetite. We call it munchies, right? Where is the problem? The problem that cannabis is not a single plant. It's a given name to hundreds of different species, chemovars, and all together in my lab only have more than 1,000 different types of cannabis. I'm the dreamer for every stoner. And every cannabis like that harbor many different combinations of these phytocannabinoids. And how to match the right flower to the right bride? This is the huge question in our field. So what I want to talk today is our approach in my lab, how to try to define which cannabis or which combination of cannabinoids will fit to a specific indication. Usually when we're talking about drug, it's a specific drug to specific indication. Cannabis is affecting all our cells in our body, actually all the system. So which cannabis, which molecule, which combination fit to which indication? So we are using different approaches, and I want to show 3 different approaches that my lab is doing. First approach, if we don't know anything, we don't have any clue if it's really affecting. So let's take whether cannabis have anti-cancer properties. We don't know that. You know, there is Rick Simpson oil and a lot of out there kind of a knowledge, but we don't really know if it's affecting. This is a huge question. So where to start? First of all, there are many cannabinoids. So when I started to work on cannabis, what we did first, we understood that we need to be able to analyze all the active compounds. So we developed methods, first of all, to analyze and to see all the cannabinoids, terpenoids, and flavonoids in the plant. We discover 11 different families of cannabinoids. Altogether, we can analyze today around 160 phytocannabinoids in the plant. In order to know how it affects our body, we did the same thing for the endocannabinoid system. So when we're taking a blood samples today or a tissue from our body, we can analyze, I will say around 100, 110 endocannabinoids and around another 40, which we call them endocannabinoids and we did a big library of the endocannabinoids. And now we are going to do the work. So if we don't have any clue if cannabis is really affecting, we're starting from a wide screen. We can grow cells in a plate. We can take tumor cells and grow them in a plate. In a plate like that, there will be between 3 million to 10 million cells, depending on the type of the cell. Cells in a plate look like that. This is a single cell. You can see in the middle the nucleus, which is a little bit less brighter. And these are cells. We can grow them in a plate, and we can grow them in a plate like that, or in a plate that have many wells. So the lower one we'll call 96 well plate. There is 96 holes there. And we can use equipment that can screen it very fast, go between the wells, take images, take measurement, doesn't matter. Don't want to go deeper on that, but can do what we call high throughput screening. Can screen very fast. And so if we're taking 96 plate and put 96 different types of cannabis or dosing, so after 24 hours, we will get from the computer that in well 9G, 95% of the cells die, whether in well, B2, nothing happened. We get the measurement. So first of all, I want to show you results about leukemia. We knew that cannabis is affecting the immune system. So we asked whether cannabis is affecting the cancer of the immune system, what we call it leukemia. So there is different types of leukemia. There is TLL and CLL and BLL. But I want to show one result about type of cancer, leukemia called TLL. So Eli Besser, PhD in my lab, took cell lines from TLL and screened many types of cancer. Every color, many types of cannabis. Every color is a different strain of cannabis, harboring different combination of cannabinoids. We screened more than 200. And you see the y-axis is viability, how many cells survive. So if the line is lower, the meaning that the cannabis killed more cells. You can see that in 2 micrograms per ml, most of the extract didn't affect these cells, but there was one buster that killed these cells like hell. So he said, okay, this is interesting. From 200 different strains, just one of them is killing very efficiently these cells. Let's see if it's something that's specific to these cells or general to leukemia. So we took 10 different types of cells of different types of leukemias, and we saw that in 50% of them, the cannabis is not affecting at all. But in 50% of them, it's killing very efficiently. So we did a genetic screen on these leukemias, and we defined that every time that there is a specific mutation or specific protein called Notch1, the cannabis will kill the cells. Every time that there is no mutation, the cannabis is not affecting. I want to take you one step backward. Cannabis is good for cancer. So we screened 200 and something types of cannabis, just one of them was efficient. For leukemia, does it affecting all the leukemia? No, just 50% of them that have Notch1 mutation. So it's very specific, okay? So Notch1 is a protein that's sitting on the membrane, and in general, I am flattening it now, the science is doing something we call contact inhibition. If you feel that there is a cell nearby, the cells won't proliferate. It sends a signal, there is cells nearby, and it's not signaling to the cells to divide. But when there is mutation, this protein is sitting awkward and not feeling the cells nearby and signaling all the time proliferate, and this is what we call driving mutation. It drives the cells to proliferate, to divide, and create cancer. So what we saw that the cannabis is affecting that. So we ask, okay, these are cell lines. Does it also doing the same thing in the mice model? So we create these tumors in the mice. You can see CT scan of the mice with the tumor, and we treat the mice with the extract. And you see on the left side, the stat of the black line is the size of the tumors with the control, which you see the tumors are growing exponentially, while in the gray line, it's the tumors that have been treated with cannabis, the mice that have been treated with cannabis. You see the size, the tumors are not growing at all. You see on the right side, the tumors, the upper line tumor before treatment, and the lower line, the tumor after 3 weeks treatment. You see how they shrink. Because this is leukemia, we did bone marrow implantation, we're creating leukemia in the mice, and we can save the mice from the leukemia. We actually did 5 different mice models, including directly cells from patients called primary cells, directly from patient to the mice. It's working all of them. But now the $100 million question, is it the entourage effect? We need all the compounds in this strain. Is it one unique compound? Is it a combination of few compounds? So these are all the active compounds in this specific strain. So we're asking, okay, how we can start to do that? So we're taking all the active compounds in this strain, all the extract, and I already can define every one of them. I know the amount of them. And if you look, if I will go one backward, you can see that part of them even doesn't have a name. 33510 or names that we gave them. Even though we know it's cannabinoid, we know the structure, nobody defined it before, so they don't have even a name. So we're taking all these active compounds, and by using equipment, we're dividing this oil, this compound, in few groups. Said, okay, I have 100 compounds, let's divide them, 25 in the first tube, 25 in the second tube. I'm fractionating the oil. And I'm coming back to the cells or to the mice with these fractions, and I'm treating them, and you can see, I have a pointer here? I have, but you don't see that. So you can see on the left side, the control, the second line is the whole extract, and then 4 fraction. And you see that just fraction 2 work as good as the whole extract. So we're already eliminating 75% of the active compounds. And we're doing this elimination back and forth, and this specific indication, we succeed to isolate 3 molecules, 3 cannabinoids, that if you take the 3 of them, 100% purified, or make them even synthetic, you get exactly the same action as the whole extract. Every combination of 1 of them or 2 of them is not working. You need the 3 of them. And we did a lot of work of biochemistry and cell biology, and I won't touch it. I will just say that in the end, we understand exactly the mechanism of action. We know these 3 molecules, which receptor they are binding. They're binding CB2 receptor and TRPV1 receptor. They activate a pathway called ceramides that cause ER stress, that elevate 3 specific proteins, that one of them is going to the Golgi, inhibiting Notch1 to go mature to the membrane. I guess most of you are not scientists, so I won't go deeper. I will just say that we understand to the bottom of the mechanism why we need 3 compounds and what is doing to the cell, that when there is mutation, the cell will commit suicide, and when there is no mutation, the cell won't be affected. Having this knowledge, this ability, I can speak very good with the physician now, with the healthcare, with the FDA, and started to take it to real clinical trials in human to prove that what we see in mice is really happening in humans. So we open a company, we raise money, and we're doing clinical trials now with this effect, which actually have 2 pathways. One, FDA approved synthetic molecules, drug, $200 million, 7 years, and gold health spree. Second pathway, we know what is the extract. In Israel, we have 110,000 patients getting cannabis under prescription under healthcare. 22,000 of them are cancer patients that getting prescription as a palliative treatment to reduce pain, nausea, increase appetite, mood. So the patient, the leukemia patient, already getting prescription to cannabis. I just need to tell him, don't take, big divorce, take white widow, which are names for medicines in Israel. I need just to fit it. So in quite fast, I can do clinical trial, what we call phase 2B, and prove whether what I see in mice is really happening in humans. So that was one way to identify what are the molecules or what are the cannabis that are affecting a specific disease. But this is when we don't have any knowledge, we're starting with an idea. And of course, I showed you what we succeed. I can show you a face of 20 miserable students that didn't reach anything, right? So the other way to go is to take knowledge that already we know. So we have 110,000 patients that getting cannabis under prescription. We have a huge clinical trial that's running outside. So what we did, we created a database on this patient. For 5 years, we follow up this patient. For one hand, every cannabis that's been picked in Israel, every plant material came to my lab to do full analysis on all the active compounds. The good things of living in very small country is that we are a small country. So we had, until 2 years ago, just 8 authorized growers that produce around 100 plant materials to the patient. So I can follow up, and I did a database on every batch, every plant material that the patient can face. On the other side, I follow up on the patient. I hired nurses. I had 7 nurses that worked under my supervision. We worked in the main clinical department in the hospitals, with the growers, in the dispensaries, and we collect data from the patient. So I can give a 3 hours talk how to spend $5 million without no results, or how to collect shitty data. And not much came out of that. But one of the things that we saw, that actually almost 50% of the patient, whether these are PTSD patient, pain patient, cancer patient, saying that the primary reason they keep taking the cannabis is sleep. 50% of the patient, whether these are pain patient, PTSD patient, or other patient, use the cannabis as a primary for sleep. So I said, okay, this is cool. Let's started to look on sleep. So we started to look on, we developed a sleep model in mice. But first of all, I want to see how the endocannabinoids play in sleep. So sleep we can see as 4 different disorders or 4 different types of sleep problems. How fast you're falling asleep? How deep is your sleep? Okay, I had a guy came to me 2 weeks ago with insomnia to my lab. He was talking to me and falling asleep and waking up, falling asleep. He don't have any problem to fall asleep, having a problem to keep the sleep deep. So it's different thing. How fast you're falling asleep, how tired you are, how deep is your sleep, whether your sleep is efficient, you're going to REM sleep, and how difficult it's for you to wake up. These 4 sleep effects are being controlled in different areas of the brain. So what we did, we took 96 mice, we divide their brain, and first of all, we looked which endocannabinoids are expressed in which area of the brain. If you're doing sleep deprivation for the mice, what is changing on the endocannabinoid? This is a paper that should be published. I won't touch it. I will just say that we learned that the endocannabinoids system play a huge role in all these parameters of sleep. So then we develop methods, or we perform methods on the mice to measure these 4 types of sleep. So we're taking in mice, and we have 24 cages. We have camera that following up on the mice. Actually, it's very unique. Mice, not unique, it's probably most of you don't know, mice are going to sleep when there is a light and are awake in the dark. So when you open the light, the mice will go to sleep. But they're first of all arranging the cage before they're going to sleep. Between 10 minutes to 15 minutes, they're arranging the cage. And it's very funny, there is a huge difference between male and female. The female will arrange the cage around 15 minutes, and the male, it's enough for 5 minutes. So as a guy that married 25 years and a father to 2 boys and 2 girls, I asked my student to do a very unique experiment to put them together. The male arranging his cage and average 10 minutes if the female is near him. Just saying. So first of all, we need to separate them, okay, between male and female. And we can measure that, how long it takes them to arrange the cage, how long they are sleeping. They are sleeping between 3 to 5 hours. And another thing, a mice is lifting his head every 2 minutes and looking around that there is no cut and put his head back. So you actually measure how tired he is, how long it takes him to arrange the cage, how deep is his sleep, how many times he's lifting his head, and the length of the sleep, okay? But here, I don't need, so, and we can add electrodes and doing EEG to measure the REM sleep, and I won't touch on that. So, in here, after I have the database, I don't need to start off a big or large screen. I already know which cannabis patient in Israel use for sleep and which are the best cannabis strain that affecting patients. So I'm already starting from a strain that I know that is extremely effective on patients. So we're taking these strains and we screen. On the upper level, you can see the red one when the mice is moving along a lot before he's going to sleep, or the lower one, it's a good treatment or effective treatment that the mice not moving much before he's falling asleep, and then we can measure that. So we took many types of strains, or not many, 12 that we know that was very efficient with the patient. We measure them and we find one strain that was consistent in every batch. You know, the growers pick a batch every 3 months. So it was very consistent, affecting the mice on 3 parameters: falling asleep, length of sleep, and how deep they are sleeping. So we took this strain, and again, we asked the question, which molecules are the one that cause this strain to be unique and affect the mice to go to sleep? So what we're doing, the same thing. We are doing fractionation. We cut it. You see on the left side a square that harboring, I don't know, 20-some phytocannabinoids. The middle one is cutting the THC alone. The right one is the rest of the cannabinoids. And we're going back to the mice. This is the left side, the middle. Just a moment, it's not working. The middle, the THC, and the right side. And we were very happy to learn that the fraction that worked was the fraction without the THC, what was the left side. So we were very excited. We have a fraction without THC, but we looked at that, and there was quite a lot of CBN. And CBN consider a cannabinoids that make you more tired or affect sleep. So we went on. We took this fraction and we cut the CBN out. And we go back and you see this is the purple one. So the blue one is the control, the red one is the whole extract, the green one is the whole extract fraction number 1, and the purple one is fraction number 1,1, without THC, without CBN, just something like 15 phytocannabinoids. So now we have a fraction, 0 THC, 0 CBN, a lot of other cannabinoids cut out, and still even working a little bit better than the whole extract. So we look inside of that, and we are going on, and we saw that there is two unknown phytocannabinoids. We isolate these two phytocannabinoids, and this is the blue one, and it's working as good as the whole extract. So now we down it to two unknown phytocannabinoids that work as good as the whole extract. We call them GTS1, GTS2, go to sleep 1 and go to sleep 2. And we are now, literally, when I'm traveling to Australia, my students trying to know whether this is one cannabinoid or the combination of the two of them, I still don't know. But what I want to say that we have now two products. One product is three products. One product is the whole extract that we already proved that is very efficient on sleep. The second product is to take this extract and to cut it in one place and to have an extract, few cannabinoids, oil, but 0 THC, 0 CBN, and still very effective. And on the regulation of cannabis, at least in Israel, this considered whole extract, you can give it as medical cannabis, and this is 0 THC, 0 CBN, and very effective. Because it's 0 THC, all the regulation in Europe, which is under 0.3 THC, this can be given. So this is a very interesting product. The third product is this molecule as a pharma, maybe synthetic or purified from the cannabis as a treatment for sleep. So I showed you two ways till now how we can approach it. First one, we don't know anything. We're doing a screen defining molecules that can affect an indication. The second one, that we already know that a strain of cannabis is very effective for a specific indication, to take this strain and to go deeper. And the third one is totally computer-based, what we call bioinformatics. So we can take from our database a specific indication, started to go deeper, but without touching the pipette and without doing anything in the lab. We can take something that we are thinking cannabis can be effective. And for an example, migraine. We know that migraine can be controlled by the endocannabinoid system. And we saw from the database, there is no indication in Israel for migraine to give cannabis. But you can be a PTSD patient that suffers from migraine. You can be a cancer patient that suffers from migraine. And so on and so on. So we saw that there are many patients that saying that when they're using cannabis, it's reduced their migraine attacks. So what we did, we sent to this specific patient, was a few hundred, a specific questionnaire about what strain they are using that reducing the migraine and dosing and so on and so on. And we divide between strains that are affecting migraines and strains that are not affecting migraines. After we had that, we compare which molecules are existing in the strain that reducing migraine and not existing in the strain that not reducing migraines. And we defined 4 molecules like that. One of them was the CA177. We went on with that and we started to do analysis with this molecule. We purified the molecule from the strain. We sent it to an we realized what is the structure of the molecules. And we started to do computer analysis and computational analysis which receptor this molecule might bind. This is all computer-based. It's not experiment. And we defined that this molecule is very effective binding a specific receptor called CGRP, which actually is extremely important in the migraine effect. And most of the drugs today that reducing migraine is affecting this receptor. So we define the cannabinoids that have a structure with high affinity to bind this specific receptor. Now we're going to the lab and we're doing this real experiment, and we see that really this molecule, these phytocannabinoids, binding in high affinity this receptor and change its activity. In order to improve that, we can take this molecule and doing small changes and increase its activity and affinity to the receptors in 100 times more. So this is not really cannabis, this is more kind of a pharma approach, but we really can see now which strains, or we can look for molecules that have this structure, these changes that we did, that maybe are existed in the plant. So this was the third approach, which is more computer-based to reach molecules that affecting cannabis. In the last 5 to 10 minutes that I have, I want to go back and look on the wider way. You know, people use plant as a medicine for thousands of years. I don't see myself as a lab that's doing cannabis research. I see myself as a multidisciplinary lab that working on medicinal plants, how they can affecting different diseases. You know, my PhD is in the molecular genetic plant biology, and my postdoc was on cancer research. And when I opened my lab, I want to do a multidisciplinary lab that's working on different plants. And usually in these conferences, I'm showing what I'm doing with cannabis, but I have 2 PhD students that working on algae, I'm working on mushrooms, and other indication. So people use plant as a medicine for thousands of years. In the last 100 years, pharma took this plant that we know that affecting specific indication. We know that if you boiling willing widow and you drink it, it reduces fever. For thousands of years, people use it. The pharma took it, isolated specific molecules called salicylic acid, did a small change, called it aspirin, and this is a medicine. And I can give tons of different medicines that coming from plants. You know, even chemotherapy, Taxol, vincristine, they're all coming from plant. But we call it single molecule, single target. Everything that was more complicated, they put aside. All the plant that was more complicated, ginkgo, chinicea, cannabis, just put aside. Single molecule, single target. The FDA is almost not approving anything that it's more than single molecule. In the last 20, 30 years, we see something that we call polypharmacology, that we are treating a disease by modulation more than one target. Usually we are using molecules or drugs that being approved as a single and combine them. If you're a cancer patient and you're going to your physician, you might get a combination of different chemotherapy. You will get C plus T plus biologic. Cisplatin that affecting the DNA, Taxol that blocking the microtubules, and the biological that affecting the receptor. You're getting a combination that each one of them was approved as a single, but you're getting a cocktail that we're creating something that doing polypharmacology. You know, the HIV virus being treated as a cocktail or different molecules that are approaching and attacking the virus from different angles. If you think about plant, or if you think about a physician 3,000, 4,000 years ago, it probably looked like that. And you came with inflammation, he wouldn't give you one medicine. He will take a leaf, a root, a flower, combine them together and give it to you. And actually what he did is polypharmacology. What I believe, cannabis opened the gate, opened a big window to start to work with more complicated plants, and also cannabis by himself, it's a very complicated plant. The difference that we have in the last 20 years called computers. 20 or 30, 40 years ago, to understand what is the complexity of 2 or 3 molecules, the pathways, the network was almost impossible. When today we can show a networking of 1,000 different proteins by pressing a button in the computer. So we are not afraid anymore to look why 3 compounds can affect leukemia. It's not even bioinformatics, it's Excel file, which few years ago was almost impossible. Today, it's very simple. So I believe that we will see more and more drugs that be more complicated drugs, and I think plants harboring that inside, and we will start to see more and more research on plants. What cannabis gave us is to laboratories like mine to have all the ability to do extraction method, to analyze these metabolites, to have different models from different disease, to do all the route from taking the plant, extract, analyze the compound, define the effect, to clinical trials. And it's opened the mind of physician, the healthcare, to more complicated action. Cannabis is actually an orchestra of molecules that working together. What we call the entourage effect, for me saying the entourage effect, it's too big, I don't understand. But this was in 1995 when Rafi Meshulam published, 1998 I think, published the entourage effect, right? Today, to show 3 molecules, 5 molecules, it's nothing. It's not complicated. So we have 160 molecules in one strain. You have around 40, 45. We can isolate, we can identify, we can start to do what we call personal medicine. Which flower can fit to which patient and how it's worked. And this issue, it's way bigger, and I need more time to explain, because a lot of the patients using cannabis to improve their quality of life. And improving quality of life, it's not one molecule or few molecules to treat a specific disease. It's affecting pain, sleep, mood, and other things. And this can be a combination of many things. But in general, when we're treating a specific disease, I think we can reach that, and I think we will face in the next few 10, 20 years, there are diseases like ALS, Alzheimer, cancer, that we hit our head to the wall for many years without any solutions, and I think more complicated treatment, polypharmacology, can solve it. What we develop in the cannabis field, in my lab, but I think there are many labs like that, it's not just cannabis, it's a platform to do this plant biology and to take it to different scale, to do extraction, analyze the active compound, high-throughput screening, define the effect, mechanism of action, clinical trials. So by that method, I will say thanks to my amazing team that doing this amazing work while I'm traveling in the world, and thank you for listening. Questions, or we done?
I've only got one word, wow. That was a brilliant presentation. Do you have the appetite for a couple of questions? Or
Sure.
not? Is that okay?
Sure.
Anyone in the room like to kick off? All right, I'm coming. Here we go. All right, so sorry, I'm coming.
Ever since you first spoke about your new profiling methods to visualize all the cannabinoids and terpenes about 5 years ago, it seemed like leaps and bounds ahead for cannabis, and the work you're doing is amazing. So thanks for coming all this way personally and presenting it to us. It's the highlight for me. I know that those methods are published, and theoretically anybody who had the wherewithal to do it could go and profile their strains in this way, theoretically. What do you think the barriers are to more widespread adoption of this deep profiling?
I don't think there is barriers. It's metabolomics, it's simple. We create the library, so these molecules were unknown in the library, but after it's known and it's there, it's everyone that have LCMS or GCMS can do that, and I guess many people doing that. I don't see any barrier. The barrier is money. It's cost a lot of money, but otherwise, I'm sure that people are doing that. I hope there is no barrier. I accept that it's not cheap. Also by HPLC, which is a cheap think today we can analyze more than 50 different phytocannabinoids. So I don't find, I literally going around the world and hear lectures, and I'm always surprised that other people not doing the same things, because, you know, if I wouldn't succeed, I would understand why they're not doing, but usually you mimic something that works. I think, you know, I will give a big compliment in Australia. I think you don't see the level of science in the rest of the world as you have in Australia. I think the Lambert Institute, we have here, and other scientists doing here amazing work, which you don't see in other parts of the world. Maybe a little bit in Canada, but you don't see the big institutes of the world stepping in, and it's a mystery for me.
Dedi, thank you again. I'm over here, speak to your left, over here this way. Hi, Dedi. Thanks again. Like the other gentleman said, I come here to listen to you speak, and it's a fascinating presentation. Thanks very much. My question is around some of the products that you are isolating, compounds you are isolating from the cannabis extracts, and I'm particularly interested in your discussion around Go to Sleep 1 and Go to Sleep 2. Are you making any efforts then to structurally identify those compounds? And then the second part of that question, in Australia, if you isolate a compound from cannabis, it's regulated differently from something that's synthetically manufactured. And so what is your intention as the next step to that? Is it to isolate those compounds in large quantities so they're actually extracts that can be supplied under that regulatory regime? Or is it to synthetically manufacture them and have them as pharmaceutical products that are regulated under that traditional model?
Okay, first of all, thank you for so sometimes people put me in the wrong place. I'm a scientist. I'm not, you know, I'm using the business side to sponsor my work, but I'm not, you know, I don't intend to do a product that doing something. I am asking questions. I'm isolating this molecule to show that they are really doing this effect. And the way to do that is to isolate them, to make them synthetic. When you make them synthetic, then it's 100% these molecules and so on and so on. But in the end, I'm asking questions that are unknown and trying to answer them. And it's going to be maybe a product and something like that, but this is out of my scope, and don't get me wrong, I don't care. Like it's, I think there are other people to do this, and I'm sponsoring my work through that, but it's not my goal. If you ask my opinion, I think there is place to both of them. I think there are patients that will prefer to take a synthetic pill with an isolate molecule and to go to sleep. And there are patients that will prefer to take whole extract cannabis as a native or natural something. And there is a patient that will prefer to take the isolate molecules from the plant. And I think there is place for everything.
I agree, but it seems to me this is such cutting edge research. There would be opportunities to partner with you or for other companies to take the benefit of your research and look at developing compounds that really can be of benefit to patients. So people in the room, if you're in the commercial space, you look at this research and partner with this brilliant researcher.
Yeah, I didn't fly 44 hours not to come with a full pocket, so.
I love it. Okay, here we go. Here's a question over just here.
Thank you. As a sleep physician, I've been looking after lots of people who haven't slept for 30 years, and they say to me, what are you going to do about it? I think what I do in my clinic, and I've got, I don't know, 3,000, 4,000, I don't know how many people I've got in my clinic. I try and characterize them, obviously, through EEG. And what you're doing is the next frontier by molecularly characterizing them, because I do think that the way forward is to personalizing the care by, well, you can be better off with a bit of CBD, you can be better off with THC and CBD, and you're better off with flower. And for us as a clinician, when we first see a patient with insomnia and you try and characterize them, the hardest bit is, do you do with a bit of CBD first, do you do with the THC first? This is why we have therapists to monitor them. But I do think your next frontier of research will allow us to personalize care. So well done on that, because at the moment all we have is the EEG. So, but sleep is a very complex subject. It's not just the lights go out and you just close your eyes. There's so many different types of sleep problems. And it still irritates the hell out of me when pharma will give you A4 sheets and say, just give them a balance 50-50, but it just doesn't work for everybody. And so some mechanism of characterizing through your research is the way forward, I feel.
So sure, first of all, we're starting to do EEG and to try that also in humans, and also in the mice we're doing more complicated. I'm not an expert for sleep. I'm working with an expert in the Technion, Professor Asya Rolls. And, you know, I couldn't run away for that because the president of the Technion, Peretz Lavi, is the world expert for sleep. So that one of the reasons I had to do sleep disorders, you know. And they're really expert, and we're doing these kind of things. But we first, in order to understand mechanism of action, at least this is my approach, I need to know what are the molecules. Because if you're using the whole extract, this extract has 12%, this plant has 12% THC, so the extract will have around 45% THC. It's masking everything. You're doing AAG and you're getting he's stoned, fine, but it's not interesting. So isolating them and you know, looking now in specific, now when I have the molecules, I'm starting to ask, what is the mechanism of action? What is affecting? Where in the brain? And so on and so on.
Sorry,
Lucy.
Dedi, I couldn't help myself. I met you in 2016 in Israel when I came over with Mike Baird, and it was a chance meeting, and I remember sitting down to lunch with you with Meiri O'Kane, the chief scientist from New South Wales, and you'd just been given your grant to start your research. And I remember you saying to me, Lucy, I'm a scientist, I'm a skeptic, I'm a realist. If I asked you now, would you still use the word skeptic in relation to cannabis?
So first of all, you know, I'm talking about this meal that we had. For many years, I'm giving this example because this was a disaster for me. I was hungry and I saw the meals going through and I had to talk, you know. Second, I'm still skeptic, okay? So I think people put cannabis not in the right place. It's amazing plant, affecting humans, affecting the endocannabinoid system. But, you know, I see videos of people are blind, taking few puffs and starting to walk. And, you know, this is too much for me. And cannabis curing cancer, I think, you know, I just showed my approach to that. What is the possibility that a cancer patient would choose the right strain to use it, okay? It's lottery. And I follow up on thousands of patients in Israel that cancer patient that took. I didn't see difference, you know, big differences. So I'm not talking about the palliative treatment, the quality of life, it's not a question. But really treat a patient, it's you know, it's a big question. After learning the endocannabinoid system and understand how wide it is, how important it is in our body, I'm not skeptical that cannabis can affect a lot of these parameters. But understanding the endocannabinoid system, the endocannabinoids, as I said, it's a mild system, it's a fine-tuned system. So in my belief, cannabis will never be the best treatment for pain. It's another tool in the toolbox of the physician. It will never be the best treatment for sleep. It's another tool for mild treatment. We need to fit which patient should use it in which situation. It's not the best treatment or everything. It's not curing the world. And when the Corona started, the COVID-19 started, the Technion said we are shutting down the Technion totally, kill all the mice, close the lab. I have Alzheimer, my mice, it's taken them 8 months to create Alzheimer. I'm not going to kill them. So I did fast 2 experiment to show that cannabis can block Corona, okay? Very easy. I'm not, do I believe that cannabis block Corona? No, I know how to do the experiment, not to shut down my lab. And the reason And the reason I don't believe, because yes, cannabis affects inflammation. I know already which molecules reduce STAT3 protein, which is important in inflammation, which reduces CXCR4, which receptors, I know deeply why it's affecting inflammation. But it will affect in a mild way. This is what the endocannabinoids know to do. Now to stop, cytokine storm in the lungs, use steroids, So I'm not skeptical, I'm more knowledgeable where it can help or not.
Okay, I've just been told we have to wrap up the Q&A. I'm so sorry, but thank you so much,
pleasure.
Dedi.
Thank
That's
you.
been fantastic. And ladies and gentlemen, unbelievable. What an incredible highlight. Such an honour to have Dedi with us today.